Thanks everybody for connecting, for the patience, and thanks for giving me this opportunity to present my work. I'd like to start with something that we are all familiar with, which is the fact that proteins need to fold in order to reach their native structure and to function, well, at least most proteins. But upon different stress conditions, a protein can get unfolded and then can adopt a different pathway, leading it to form these amorphous aggregates or even these amyloid fibers. And you can see from this energy landscape that these amyloid fibers are very stable, they're very low in energy. And the problem is that they're linked to a number of neurodegenerative diseases. So you can see some examples here on the slides. The good news is that we have machinery that can counteract this. And these are molecular chaperones. And it was recently identified that HSP70, a molecular chaperone, together with its co-chaperone, the J-domain protein, can reverse this process and disaggregate amyloid fibers. And the way these proteins work together is follows. The J-domain protein recognizes substrates or clients. This can be unfolded proteins or misfolded proteins. It brings them to HSP70. The J-domain will hydrolyze the ATP hydrolysis to ADP. HSP70 will get locked onto the substrate, and upon ADP release and new binding of ATP, the substrate can get released and undergo its correct, and it can refold into its correct structure. But this can't really explain fiber disaggregation. We don't have this bind and release going on in fibers and not this refolding either. And trying to decipher this mechanism of how fiber disaggregation works, we can see a hint that it's pretty established that only one class of J-domain proteins can assist HSP70 in fiber disaggregation. So to have a closer look at these J-domain proteins, they consist of two main classes. There are more, but these are the ones I will focus on, class A and class B. And you can appreciate that they share high sequence and structure similarities. They both consist of a J-domain. This is the domain that enhances HSP70 ATP hydrolysis, the CTDs. These are the suggested substrate binding domains, and they're both homodimers. This is the dimerization domain. Class A has a zinc finger-like region, and they both have a glycine phenylalanine-rich linker. And you can see here the structures of only the CTD regions. So these J-domain proteins, or JDPs, or HSP40s, you'll see them in the literature, are very similar, but these classes are not the same. And like I said, only class B JDPs can disaggregate amyloid fibers. And we wanted to find out what exactly is this fiber disaggregation mechanism and how is it related to the difference between the two classes. And if we look at the cycle, we know that JDPs will interact with substrates and with HSP70. And since it's something that works on the fibers, this disaggregation, we check the interaction with substrates, with preformed amyloid fibers. And we saw that class B and class A interact very similarly with the amyloid fibers, so this can't be it. And then we thought maybe there is something more to the interaction with HSP70. So it's known that the J-domain is a part that enhances the ATP hydrolysis of HSP70, but maybe there is something more that is different between the two classes. So with this in mind, we took our J-domain proteins, and this is our class A representative DNA JA2. We took out the substrate binding region, so we're left with the J-domain and the linker. This is N15 labeled, and we recorded in HSQC. When we added HSP70, we could see selective peak broadening, and after assignments, we could map this onto the structure, and we saw an interface here between helix 2 and 3. And this matched previous knowledge. This interaction is already characterized and very well conserved. When we did the same for our class B representative, DNA JB1, we added HSP70, but to our surprise, we saw no binding. And this really puzzled us. When we took the shorter construct of only the J-domain, we saw binding, a nice selective peak broadening, and when we mapped this, we saw the same interaction surface on between helix 2 and 3. So what was going on with this longer construct, and what is this linker doing here, and why does it prevent the binding? And this is the point to mention my friend and my colleague, Meital, who did all the structural analysis in this project, and together with assignments and RDCs, and PREs, and Talos, we were able to analyze the structure, to solve the structure of this extended region. And we saw that the GF linker isn't just a disordered linker, but actually it contains a fifth helix, which docks onto helix 2 and 3, preventing binding to HSP70. And this exists only in class B and not in class A. And now we were curious to see then what goes on in the full-length protein. So we took our class B protein, DNAJB1, and this is a homodimer. Like I said, it's 80 kilodaltons about in size. So here we used selective methyl labeling. We labeled isoleucine, leucine, valine, and methionine residues, and you can see here the carbon-proton correlations. And when we added HSP70, we could see selective peak broadening, indicating binding. And when we mapped this out, we saw that it's located on the J domain, as we expected, but also on CTD1. We did the reverse experiment with HSP70, and we could see that the binding site for CTD1 is this EVD tail. So it's the last 4 residues in HSP70, EEVD, and the binding site for the J domain is what was already known here. And this set of interaction between CTD1 and the EVD doesn't exist in class A. So we thought if this is something that has to do with the difference between the two classes, what happens if we take out the EVD tail? So we made a construct without the EVD tail called HSP70 delta EVD. We added it to our full-length protein and we saw no binding. So there's no binding to either site on DNAJB1, even though the other binding site is still here. So what's going on here? Why is the J domain inhibited in the full-length protein? And more importantly, how is it released? And is the EVD tail the key to the release? So if so, what would happen if we added only the tail this time? So we generated a peptide corresponding to the tail. We added it to DNAJB1 and we see shifts in residues from CTD1. So this is the binding site that we already knew for the tail. But looking closely at our spectrum, we also saw a new set of peaks appearing adjacent to J-domain peaks. So here are 2 examples marked with an asterisk in isoleucine 20 and isoleucine 48. When we add the peptide, we see a new set of peaks forming. And in order to find out what these were and what was going on in the full-length protein, we carefully compared our spectra. And we saw that in the full-length protein, and like in DNAJB1, if we look at isoleucine 48, it has the conformation of the isoleucine 48 in the J-domain GF construct, meaning it's inhibited closed conformation. When we added the peptide, we saw a new peak forming. And when we wanted to find out what this peak was, we compared it to the free J-domain, and we saw that indeed, this is the peak of the free J-domain, so the same residue in the conformation that is free and able to bind HSP70. So addition of the HSP70 tail, the EVD tail, frees the J-domain and enables subsequent binding. So our model for interaction is that if we take our class B protein, the DNAJB1, it has 2 binding sites for HSP70, but one of them is inhibited. And when we add HSP70, it first binds through its EVD tail to CTD1. The inhibition is then released, the J-domain is free, and then it can bind HSP70 and activate it. In class A, there isn't another binding site, and there's no intramolecular inhibition. The J-domain is always free and able to bind. So we've noticed this difference of interaction between the 2 families, between the 2 classes, but what is it good for? And I started by telling you that only class B can disaggregate amyloid fibers. So only class B can assist HSP70 in the disaggregation, while class A cannot. And we thought that if we now found out the difference in the interaction with HSP70, it might have to do with the two interacting regions. So if 2 binding sites are necessary, we thought maybe we can generate some super efficient co-chaperone. And we generated a mutant that has 2 binding sites for HSP70, just like DNAJB1, but no intramolecular inhibition, like DNAJA2. So kind of like a hybrid. And we were expecting this one to be very efficient in fiber disaggregation. But when we added it to HSP70 into fibers, we saw that there was actually almost no disaggregation. So it performed very similarly to our class A. And in order to figure out why, we were lucky enough to have our close collaborators, the Bernd Buchau lab in Heidelberg, and specifically Anne Benting, decipher the mechanism of amyloid disaggregation. And in this beautiful paper that they published, you can see how DNA JB1 binds fibers as a substrate and then recruits HSP70 to the fibers, and it recruits multiple proteins to the same area. There is clustering of HSP70. This generates enough pulling forces in order to have some piece of fiber all move out, and this is the fiber disaggregation. And when we look at each of these steps, we wanted to see if there is a difference in the fiber binding of our different JDPs. We took our class A, our class B, and our hybrid mutant, the delta helix 5 with no inhibition. And we saw that they all bound fibers very similarly. When we tested the recruitment and the binding to HSP70, and I'll show you here the binding, we can see also that they bound fibers very similarly, and class A also recruited HSP70s very similarly. But when we wanted to check the clustering, and we did this with labeling HSP70 with donor or acceptor dye and monitoring the FRET efficiency, we saw that only class B wild type could cluster HSP70s on the fiber. So class A couldn't, and our mutant hybrid couldn't either. So it's not only the two sites that are important for the fiber disaggregation, but it's also the intramolecular inhibition that is crucial for clustering HSP70 onto the fibers, which enables the fiber disaggregation. And now that we know how class B specifically aids HSP70 in fiber disaggregation, we can now go on and to investigate what goes wrong in disease and how different mutations which alter the inhibition can affect. And with this, I would like to conclude, and I'd like to thank all my group, and specifically Meital for all the work on this project, the Buchau group with Anne Benting, Nadinat, and of course to you for your attention.
Thank you very much indeed. That was great. So save your questions for the breakout room. We're going to move straight on to our next speaker, which just happens to be me. So I'm going to share my screen. Okay. Right, well, thank you to the rest of the organising committee for allowing me to speak today. I'm going to be speaking about NMR structure validation. So validation is important. You know, structures are only useful if they're accurate, and that we can show they're accurate. So if you go to the PDB and have a look at any X-ray structure, you'll find a nice mix of model versus data validation, which specifically validates accuracy, as well as geometrical validation, which validates the quality of the geometry. You also find some metrics which can tell you if the model's been overfit or not, and you've also got the resolution. So overall, in general, X-ray structures are very well validated and they're quite useful. In contrast, if we go and have a look at an NMR structure, we only find geometrical validation. And of course, this is the problem because a protein can have very nice geometry, but that doesn't necessarily mean it's accurate. Another slight issue is that NMR structures tend to come as bundles of 20 models. And there's good reasons for that. But to anybody outside the field, this can be a bit confusing. For example, a bioinformatician might come along and go, oh, there's 20 of these, why? Which one am I supposed to use? And I know that's the case because I was that bioinformatician. And unfortunately, the standard approach is to generally not use any of them and to completely ignore NMR structures. And again, I know that because, well, that's what I did. So this is a problem. We need new validation tools to improve confidence in NMR structures and to encourage the wider community to use them. So I'd like to present our new method for validating NMR structures, ANSWER, which stands for Accuracy of NMR Structures Using Rigidity and Random Coil Index. So briefly how it works is we compute the flexibility of the protein using backbone chemical shifts, and then compare that to the flexibility of the structure that we compute using rigidity theory. And the idea is if these two things agree with each other, then perhaps we have an accurate structure. So here's some example output for this particular protein. It doesn't really matter. So in blue, we have the flexibility according to our chemical shifts. And in orange, we have the flexibility according to rigidity theory from the structure. And what we'd like to say is, well, this region is looking, pretty okay. whereas over here something funky seems to have gone on, and maybe we want to look into that in more detail. So what answer then does is it computes the correlation and the RMSD between these two measures to get a score. So rather creatively, these two scores are called RMSD score and correlation score. They're out of 100, 100 being the best. They're essentially percentiles. And you can plot both of these scores on a 2D plot like this, so that the best scoring structures will appear somewhere in the top right-hand corner here, and the worst would be in the bottom left. And you can see here for this example, these measures seem to agree quite nicely and have two high scores. So towards the end of last year, we published the method, and one of the first things we talk about in that paper is refinement in explicit solvent. So this is one of the last things you'll do when solving an NMR structure, when solving an NMR structure. And you do it in recognition of the fact that experimental restraints are not sufficient by themselves to fully resolve all the local detail, for example, hydrogen bonds or hydrophobic tethers. So refinement is essentially a short simulation to improve the local geometry. And importantly, the NOE violations, your distance violations, which is the main validation you sort of use as you proceed in the calculation, they don't change very much, if at all, during refinement. So therefore, according to NOE violations, the structure before and after refinement are of the same quality, the same accuracy. But of course, they're not. You know, we accept that the refined structures are better. That's why we do it. So the question is can answers spot this improvement in accuracy? And the answer is yes. So here's an example of an alpha helical protein. So I'm pointing out it's red means alpha helical. Never mind. In grey, we have the flexibility predicted from shifts. In blue, we have the flexibility from the structure before refinement, and in orange, after refinement. And you can see that after refinement, the flexibility is in much better agreement with the flexibility predicted from shifts. And this is across the board. The whole protein has become more rigid and more correctly rigid. Here's another example for a beta sheet protein. And again, we see the same kind of improvement. And then here's just one more example, which happens to be this protein over here. So let's take this a bit further. So I was using answer to compare NMR structures and X-ray structures. And one thing I've noticed is that X-ray structures tend to be a little bit too rigid. And I guess this makes sense if we consider that X-ray structures are solved in crystalline arrays and often at cryogenic temperatures. So we wouldn't necessarily expect the flexibility of these structures to match that predicted from shifts attained in solution and at room temperature. So I suggested that perhaps X-rays solved at room temperatures should have better answer scores. And that's exactly what we saw. So here I've got 4 structures of lysozyme solved at cryogenic temperatures and some others solved at room temperature. And we can see the room temperature ones score better. At cryogenic temperatures, the structure has peaks in flexibility missing that the shifts suggest should be there. Whereas at room temperature, these peaks start to appear. And again, I'd like to point out that just overlaying the structures makes it quite difficult to see these differences, because these differences can be in the local details, such as hydrogen bonds. Although we can see some slight differences between loops for the cryogenic and room temperature structures. So since publishing that paper, I've computed answer scores for a big chunk of the PDB. So one of our main results is that the accuracy of NMR structures has gradually increased up into about 2005, at which point it's kind of just leveled off. And we think this is because the sort of general way of solving NMR structures was established at around or just before this time. For example, the paper on refinement in explicit solvent came out in 2003, as did 2 programs we still use today for solving structures, Cyana and Explore NIH. So I've just uploaded a preprint on bioRxiv, which looks at this data set in more detail. And to go with it, I've launched answer.com, which is a website which has pre-computed answer output for nearly 7,000 ensembles. So if you have a favorite NMR structure, you can go to answer.com to see what Answer has to say about it. So what am I doing now? Well, currently, I'm looking into whether Answer can be used to predict the accuracy of decoy protein structures. So these are kind of physical, plausible structures which represent the range of accuracy of structures generated by structure prediction programs. So here I've got the 16,000 decoys and their predicted accuracy according to Answer and their actual accuracy. And we can see that Answer does a fairly good job of this. But I've always suspected that answer scores might be somewhat dependent on the type of structure being validated. For example, an alpha helical protein might by default score better than the beta sheet protein. So to look into this, I started playing around with deep neural networks. And I found that actually, if I just put the answer scores into a deep neural network, then the prediction of decoy accuracy is improved. And then if I start to feed in some other features such as size or secondary structure content or the average flexibility, then these predictions continue to increase. So what I'm hoping is that the network is learning how to account for different types of proteins, and that this might be a way to improving answer as a validation method in general. I'm also interested in combining answer with other validation methods because, of course, no validation method is infallible. So a PhD student in our group, Mariam, has shown that building an ensemble using models with both good answer scores and good energy leads to an ensemble which has better Ramachandran angles than an ensemble built using just answer scores or just energy. So I want to take this a bit further. I want to combine many different types of validation to try and work out the best combinations. And again, I think machine learning would be a good way to do this. Mariam has also found a rather nice way to combine answer temperature coefficients and cyana to improve the hydrogen bonding network in structures. And what's nice here is we see improvements in answer score as we add hydrogen bond restraints, but also as we remove those that we have less confidence in before we go on to refine. So hopefully I've convinced you that answer is a useful method for validating NMR and structures. So if you're interested and you've already deposited your structure to the PDB, then you might be able to find the results, the answer output for your structure on answer.com. Alternatively, you can download the software from my GitHub account, currently only installed on Linux machines. But we also have a version of answer working on NMRBox. I'm not sure if it's been released to the public, but it should be soon. And I'm also going to be working with CCPN so that the answer output can be read into the CCP NMR analysis if you use that. And of course, if you have any questions or if you want to talk about your answer output for any particular structure, please let me know. So I'd just like to thank my boss, Mike Williamson at the University of Sheffield, one of his PhD students, Mariam, for testing answer, as well as our collaborator in Japan, Adnan, the BBSRC for funding the project. And if you're listening, BBSRC, please fund our grant. Not sure if that will work. And of course, everybody here for listening. Thank you very much.